Hey guys, can you hear me? Yeah, okay, good. I'm having some internet issues on my end. So I think I'm gonna start class with the camera off to see if that helps resolve some of the issues I'm having. So I'm just letting you know that. Thank you. Yes, I'm feeling better. Thank you, Natasha. It's been a very stressful week and it's gotten the better of me sometimes, and not enough sleep, not enough self-care, right? So but no, I'm feeling much better. Thanks for asking. Sorry about the camera issue, but I'd rather keep the camera off for now in hopes that we can get through the rest of the material so that you guys can go and have a great spring break, and that we don't have too much lag time here. So let me share my screen. Okay. Can you guys see the PowerPoint slide? Thank you. Okay, thank you guys for answering. Everyone else is on their phones, I guess. All right, so let's finish this out. So last time when we were talking about the ferns, we finished up with talking about how the sporophyte generation shoots their spores, and I put some videos on online for you guys to watch that. And so those spores here, let me just see here. Yeah, and so those spores are going to land on the ground somewhere and grow into the gametophyte generation, right? Because the spores grow into the gametophyte generation. And you might remember from the very first class, or when we first learned about alternation of generations, that we use the fern as the example plant, because it's nice because you can easily see the sporophyte and the gametophyte generation with your eyes, and so it's easy to see all the parts. But anyways, here's the gametophyte generation of most ferns. Very simple, thallus, heart-shaped, etc. Since it is the gametophyte generation, we know that there's going to be sex organs on it, because gametophyte generation makes gametes, egg and sperm. And so if the fern has a gametophyte that looks like this, the archegonia, which have egg inside, are going to be located near this little notch. And the antheridia are going to be kind of like spread out. So in a way, we've already gone through fern alternation of generations in that second lecture, when we were first talking about alternation of generations in general, because we used ferns as that example plant. So I'll go through this quickly because it has a lot of the same players we're used to dealing with, right? On the left here, you see the adult sporophyte plant. Oops. Let me use, right? On the left here, you have the adult sporophyte plant. It has all the things on it that we would expect of a fern. It has leaves, which are now called megaphylls. It has clusters of sporangia that we call sori, horizontal stem, real roots, and the young leaves are going to emerge as fiddleheads, right? So if we were to zoom in onto one of these, one sorus, we would see clusters of sporangia. And if we were to zoom into one sporangium, right? This is it here. We would see the spore generating tissue eventually, because when the sporangia are mature, that annulus that we talked about before, that special layer of cells, will pull back and allow the sporangium to shoot the spores out, right? And that's what the videos that I posted show you guys. So here you go, the spores shoot out. The spores are now haploid, right? Because they are the result. I gotta move this box here, right? The spores are the result of meiosis. So they're haploid. And they're gonna shoot out. And because we're dealing with notice here, it just says spore, right? And mature gametophyte plant. So we're dealing with a homosporous plant, right? Because we only have one spore type. Majority of ferns are homosporous. We'll talk about this a little bit more as we finish today's course, today's content. But notice spore. So we're dealing with a homosporous sporophyte plant, which means the gametophyte plant is going to grow outside of the spore wall and usually contain both sex organs on the same plant. And so it does. You could actually see it here. Look, germinating spores. Here's the spore wall. Here's the spore wall. Here's the spore wall. Notice the gametophyte plant growing out of it. Here. You can see it very well in this one. Here's the gametophyte plant. Whoa, growing out of it, right? Growing exosporically. So on that gametophyte plant, you're going to have male and female sex organs. Those male and female sex organs will have egg or sperm and egg respectively. Eventually sperm will leave, right? Here's sperm leaving. Sperm will leave and swim, 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 fertilize the egg, shown here, fertilization. And when haploid sperm and haploid egg fuse together, the result is your 2N zygote, right? 2N zygote. 2N zygote, diploid zygote, that's going to grow, diploid zygote, going to grow into the new sporophyte plant, right? Notice the color change in green here, right? So diploid zygote, going to grow via mitosis and cytokinesis, grow, eventually into that new sporophyte plant. Just so you can see it here, right? Obviously, let's go back. Obviously, this zygote, right? I'm really trying to hit this home because there's still confusion. Just because still confusion. Look, this egg, where is this egg, right? Where is this egg? This egg is inside of the gametophyte plant, right? In the female sex organ. So when that egg is fertilized by the sperm, the new 2N diploid zygote is still attached to that gametophyte plant because that's where the egg was, right? Egg stays. Egg stays. So as the sporophyte generation grows, it's initially attached to that gametophyte plant because that's where the egg was. And so sometimes you'll see something like this. So I'm using orange to do the gametophyte generation. And I'll use blue. So here, look, this is the new sporophyte plant is emerging. That's the shoot system. And here's the root system of that new sporophyte plant developing out of that gametophyte plant because that's where the egg was. Eventually, the gametophyte plant eventually the gametophyte plant will just disintegrate and go away because why? Because now the sporophyte plant, how do I get back to draw? Clear all drawings. Let's just see. Oh, here we go. Yeah. Because now the sporophyte plant has a leaf, so it can do photosynthesis. It has a root, so it can get water. doesn't need the gametophyte plant anymore. And so when you get to this point, the sporophyte plant is all on its own. It's not attached to the gametophyte generation anymore. And it's dominant. It'll be the generation that the plant exists in the majority of the time. Okay, so obviously we need to go to the next one. So let me share this. Yep. And this one won't, I know I say this sometimes and it ends up not being true, but this one it won't take that long to go through. And we'll talk about the phenology assignment mentioned in the syllabus today too. Okay, but we're just going to talk about a few different genera of fern. And like I said, we'll talk about that phenology assignment as well. So here's our good friend Fern, right? Hanging out in the temperate forest, doing its ferny thing. And remember back in that first lecture, we saw this, right? And this is that biological taxonomy of how we categorize or order living organisms so that we know who's who, right? So we can identify species. So some of these we've talked about more than others. For example, in the first lecture, right, the domain Eukarya, right? We know we're in the plant kingdom, right? We've covered a lot of phyla, right? A lot of phyla. Right now we're in the ferns, Polypodiophyta. But we haven't really talked much about class, order, or family. And so today, just so you know, we're going to cover a few different orders of ferns. And that's what I mean by orders. So here are some orders in the phylum Polypodiophyta. You have Filicales, Marsileales, and Salviniales. That's how you say that. When you see this ending in the plant world, oh, okay. When you see this ending in the plant world, ales, that's how you say that, ales, just so you know. Filicales, Marsileales, and Salviniales is the orders we're going to talk about today. Most members of ferns are in the order Filicales. So if you were to make a pie chart of all the ferns, right, that's what this is, a pie chart of all the ferns, the green slice, which actually in this case is the majority of the pie, right, the green slice is Filicales. 80% of all the species are in this order. And in Filicales, all members are homosporous. So that alternation of generations that we just went through would be an example of a member of Filicales. And that's why we use that example to describe the alternation of generations in ferns, because most members are in this order and have that homosporous type of existence. And you notice here, there's 22 other orders though, right? We're only going to talk about a total of 3. Okay. Yeah, we're only going to talk about a total of 3. But just so you know, there are 22, at least 22 other orders of ferns. Okay. Who's in Filicales? So here are some highlighted species in Filicales. You'll be able to see at the very least herbarium specimens of these species in lab after the break. So the reason I highlight some of these species is because, number one, they are found in your area. So if you're hiking around during break, you might see some of these starting to emerge. And if not, then this summer when you're walking around, you may see them. So that's one reason. Another reason is because it's they're a good representation of like some of the different structures we've already talked about. So here's the first one. This is the common polypody. Just so you know, if I ask you any questions about this on an assignment or on an exam, the common name is fine. The common name is fine. And in many cases, I'll put both next to each other, common name and the species name, just so you know. So you don't have to memorize Polypodium virginianum. Common polypody or rock polypody is fine. All right, so here is that polypody, right? And notice it's Polypodium, so this is one of those species who's in that genus that the phylum's named after, right? Polypodiophyta. This likes to grow on rocks, as the common name suggests. So if you go up to Palisades, you might see them there. On the left picture is the top side of a leaf. On the right picture, or in the center picture, hold on, excuse me. One sec. On that left picture is the top side of the leaf, and that center picture is the bottom side of the leaf. Notice, what would be a good color? I guess white. Notice on the bottom side of the leaf, you see these green clusters. Those are the sori, right? Each one being a cluster of sporangium. Here it is, growing in next to a rock. Next is sensitive fern. Sensitive fern is called sensitive fern because if there's a frost, it's very sensitive to frost, and so the leaves will immediately kind of like turn yellow and die away. That's why it's called sensitive fern. Sensitive fern has two types of leaves on each plant. It has a normal looking photosynthetic leaf. And then it has leaves that are modified to only bear sporangia. And this is something that we call leaf dimorphism. I'll write it in a chat here. Dimorphism. Remember, di means two and morph means forms. So sensitive fern has two leaf forms, right? One on the left here that's photosynthetic and regular looking, and the photosynthetic one does not bear any sporangia. And then one leaf that leaf type that bears all the sporangia on it. So a little bit different than what we're used to looking at. Oh, but if you walk around in the woods this time of year, when everything's still kind of not awake yet, you'll see these sticking up. In fact, they use these sometimes in flower arrangements even, because they dry very well. All right. Next is Christmas fern. Christmas fern on the left, that's the whole plant. In the center photo here, you're seeing the underside of the leaves, and notice all of the sori. Christmas fern, the way I remember Christmas fern is that the pinnule, and if that term, or sorry, in this case, it would be pinna. If that term seems unfamiliar, go back to the other recording on the ferns. But Christmas fern, I remember as being Christmas fern because the pinna looks like a mitten. I don't know if you know what a mitten, not a glove, right? A mitten, right? Like here's the thumb, and then here's the rest of the, be for a very long hand. But that's how I remember it being Christmas fern. Here it is in nature. You can see all the sori on the underside of the leaves. Next is maidenhair fern. Maidenhair fern is one of the type, this is a good example of why sometimes even botanists just give up because this is one leaf, and like how many times pinnately compound is this, right? It's like, oh, it's too hard to tell. You would have one thing here, then this thing would be coming off here, this thing coming off here, this thing coming off here, etc., etc. So this one we don't even talk about it, about how many times pinnately compound. We just say it's very compound leaf. Maidenhair fern is something you would see a lot of times planted in botanical gardens, particularly in more of like the Zen garden areas of the garden. And maidenhair fern has an example of what's called a false indusium, where it doesn't have a special membrane that protects the sporangia. Instead, the sides of the leaf, or in this case, the sides of the pinnules, bend over, like kind of curl over the sporangia to protect them. Serves the same function as a indusium, but it's just an example where it's only the leaf curling over, not a new membrane. If the word indusium, here I'll write it. Indusium, plural, indusia. If this word looked unfamiliar to you, go back to the last lecture. Okay. Bracken fern. Bracken fern, the only reason I bring it up is because the leaves are like, can be up to like 5 to 6 feet tall. So if you do see a herbarium specimen of it in the lab, you'll probably only be seeing like this part, this portion of the leaf, because the leaves themselves can be like I said, like 5 to 6 feet tall. Bracken fern is another one that has this example of this false indusium. Here, the edges of the leaf just curl over uniformly, and the sporangia are located underneath those. Then we're going to talk here about 3 different types of Osmanda. You have Osmanda cinnamonia, so the cinnamon fern. All 3 of the Osmanda plants we're going to look at right now have this dimorphism. Osmanda cinnamonia, or the cinnamon fern, has regular leaf dimorphism, again, just like the sensitive fern. And just like the sensitive fern, same deal. It has some leaves. Oops, where is my annotate? Oh, and what would be a good color here? I guess black. Yeah. So some leaves are just green and regular looking and photosynthetic. Other leaves are specialized in that they have only sporangia on them, right? So another example of leaf dimorphism, just like the sensitive fern. Here's a close-up of one of those sporangia bearing leaves. And you can kind of see where cinnamon fern gets its name, right? The leaves bearing sporangia kind of look like a cinnamon stick. And after a frost, when the leaves are going to die back for the winter, this is the color that the plant turns, which is also kind of a cinnamon color. So leaf dimorphism here, just like we saw in sensitive fern. And this next member of Osmanda is this interrupted fern. Here we have dimorphism of the leaflets, right, or the pinna, right? Oops. So here we have, oh, geez, text, right? Black. There we go. So here we have pinna or leaflet dimorphism. Now it's not 2 different leaves like as we saw in the previous cinnamon fern or in sensitive fern. Now on one single leaf, you have dimorphism of the pinna or the leaflets. Let me move this here. So for example, oh, I guess I can't do both things at once. All right. So here is one leaf. And on the top part of the leaf, I'll use blue. On the top part of the leaf here, and you can't really see it, but kind of at the bottom of the leaf too, the leaflets or the pinna are just green and regular. See here, kind of green, regular pinna that do photosynthesis, and they don't have any sporangia on them. However, in between those sections, or interrupting those two sections, I'll do pink, are leaflets that only have sporangia on them. Hence, hence the name interrupted fern. So the fertile pinna interrupt the leaf in the middle. See, I can draw it again on this one here, right? Green section down here, green section up here, and then the fertile pinna interrupting that leaf. Remember, this whole thing is one leaf, right? And last but not least, oh, here you go. Here you go, showing you in, oops, in nature. Showing you in nature. Let's go here. Right, here is the green part on the top. Here is the green part on the bottom. And interrupting this leaf is those fertile leaflets or pinna. Okay, last but not least, this is another member of Osmanda. This is the royal fern. Royal fern has regular, or has also leaflet, or I should do it the other, I just do it the same way, pinna or leaflet, leaflet, yeah. Dimorphism, just like interrupted fern. However, here it's a little easier to see because what ends up happening is the fertile part is going to be at the top of each leaf. So fertile part, oh, I don't know why I did that. I don't know why I made it pink, but that's okay. Fertile part is going to be the top of the leaf, and the non-fertile green part is going to be the bottom of the leaf. This section here doesn't have a fertile part yet, but the fertile part would eventually come, like would be this part up here. And what we say to remember to keep this common name here, royal fern, you can say that the royal fern leaves have the sporangia bearing portion on the top like a crown. So these pictures may not do these plants justice. So what I do, I have this drawing here that maybe makes it a little easier to see. These are those three Osmanda plants that we just covered. So going backwards, here on the right is that royal fern. This has pinna dimorphism because these pinna are green and photosynthetic, but these pinna are fertile and just bear sporangia. The interrupted fern next to it also has pinna or leaflet dimorphism because these leaflets are green and photosynthetic, and interrupting them are these pinna that have sporangia on them. But the cinnamon fern has just regular leaf dimorphism because you have one entire leaf that's green and photosynthetic next to other entire leaves that bear sporangia on them. So it makes it a little clearer. This is so when you're studying on your own, kind of get an idea of what I'm talking about. Right, we're just going to briefly talk about a couple members of these other orders. So Marsileales and Salviniales are examples of ferns that are in these 22 other orders, right? So that's where they are in this chunk of the pie. And if you were to look only at that chunk of the pie, if you were just to pull, like if this was a pizza and you pulled that blue slice off and we're looking just at that blue slice, here's what you would see inside of it. So Marsileales and Salviniales, they don't make up a lot of that slice, but they're more common than some of the other orders, and so that's why we talk about them in this class. Right, so Marsileales is only 3% of that slice, Salviniales is only 1% of that slice, and the 20 other orders that we do not cover in this class make up the 96 other parts of that slice. Okay. Both Marsileales and Salviniales are aquatic ferns. So for Marsileales, they are emergent aquatic ferns. This is a word we covered during the horsetails. Let's see, what would be a good color? I guess white. Remember, emergent, covered that term in the horsetails. It means the plants grow in water, but they a lot of times stick out above the water. Sometimes if the water gets high, they might appear to be floating plants, but they're attached to the bottom. And look, important, all members of Marsileales are heterosporous, which is different than all the ferns we've covered so far that were in Filicales, right? So what do I want you to know about Marsileales? I want you to know it's an aquatic fern. I want you to know that all members are heterosporous. And the sporangia, they also have this kind of dimorphism in that the leaves or fronds that have the spore-bearing structures on them are called a sporocarp. I don't really care too much about a sporocarp. Here is an example of a member of Marsileales. This is water clover. You can see it does look like a four-leaf clover, right? If you were to see this growing somewhere, you might think, oh my gosh, it's a four-leaf clover, right? Yeah, it's actually a fern. But it had if you saw this growing, you probably would think to yourself, that I don't know what that is. That doesn't look like a fern to me, Rebecca. True, but look at this dead giveaway, right? Fiddlehead. So we know it has to be a fern because only ferns have fiddleheads. Here is this is what I was saying on the previous slide. Just like some of the other members we looked at, this water clover has, again, leaf dimorphism. I'll write it again. Leaf dimorphism. Oh, not lead dimorphism. Leaf dimorphism, 2 types of leaves. And one leaf that is green and photosynthetic, and the other leaf that's kind of modified to bear the sporangia, and that's what was mentioned on the previous slide is the sporocarp. There you go, sporocarp. Salviniales, also aquatic ferns, also heterosporous, and same deal, sporocarp, right? So what do I want you to know about Salviniales? They're aquatic. Oops, they're aquatic and they're heterosporous, just like Marsileales. Both have the same thing. Both have sporocarp, everything the same. Here is Salviniales. Believe it or not, this is also a type of fern. This is Azolla, which is a type of fern that the common name is duckweed, although a lot of things are actually called duckweed. So actually we'll see another plant later on in this course that's also called duckweed. Two different types of plants. But anyways, this is Azolla, very, very, very small plant. Again, aquatic plant. Look how small. This is one of the ones when I was talking about different sizes of ferns. This was one of those like super small examples. Look, this is a person's hand grabbing a whole handful of these plants. But even though they're super small, they'll have the same characteristics of ferns. They'll still have the leaves emerge as a fiddlehead, things like that. Azolla has a symbiotic relationship with cyanobacteria. And the cyanobacteria that it has the relationship with is called Anabaena. But basically, it's similar to what we saw in the hornworts, in that here, the plant is providing a nice home for the cyanobacteria. And as a mutual kind of symbiotic relationship, the cyanobacteria is going to provide nitrogen for the plant. So on the left here, you can see the scale, right? This is 2 millimeters scale. That's your Azolla. Now we're at 500 micrometers. Look how, you know, this is really, really small leaves. This can't, oh yeah. And then it says 10 because we're under the microscope. But this thing on the right here is that Anabaena, that cyanobacteria, Anabaena. I think I have, yep. Just so you know how this all works, because you're like, how does the bacteria like live inside of the plant? So if you were to like look at one of these Azolla leaves, a cross section under the microscope, there's an entire hole inside of each leaf where the bacteria live, which is pretty interesting. The bacteria must be very important to this plant, or else why would the plant dedicate so much of its leaf space to a home for this bacteria to live in? So that the thing I circled is, you know, that's in a drawing, but here it is under the microscope slide. So if you like did a cross section through a leaf and then you saw inside this whole cavity filled with these darker dots, those darker dots are that cyanobacteria. So pretty cool how things like this evolve, right, over evolutionary time, and it benefits both organisms. Another type of Salviniales is Salvinia, named after the order. Water moss. Water moss, also super tiny. You can see it here on this person's finger. Water moss also has leaf dimorphism, but here it's a little weird. Water moss has lost some parts of its plant body over evolutionary time, and meaning roots, no more roots. If you think about it, it's floating in water, right? So it really doesn't need roots. It's surrounded by water. So it no longer has roots on it. Instead, it has a stem, which you can see here. And then dimorphism in leaves again, leaf dimorphism, in that you have 2 green floating leaves. So here's one here, here's one here. Those are what's going to do photosynthesis. And then one of the leaves has actually lost its green tissue, or its mesophyll. It's lost that over evolutionary time, and instead you just have the veins of the leaf left over. And if you want to say, what does that do? You can kind of say, yeah, fine, it serves as, I guess, roots, but it is floating in water. So, and then this weird thing here is where the sporangia are located, and they're located on this weird dissected leaf. Yeah, weird, super weird. Okay. The water moss is fun. If you ever get your hands on some water moss, it's fun to play with because the leaves are covered. I don't know if you could see it here a little bit on the picture on the left. But basically, the leaves are covered in these trichomes, these little hairs, and the little hairs attach to bubbles. And so the plants literally cannot sink. So you can like put them in water and try to use your hand to like splash them down and like get them to sink, and they won't sink because the little trichomes catch these air bubbles and keep them afloat. Both the we're still in Salviniales here. Both Azolla, or the duckweed, and the Salvinia, the water moss, are problematic invasive species. And I just want to make clear here, because sometimes it annoys me. So invasive means a plant or a species that is very aggressive and competitive and able to kind of overtake resources compared to other plants. Invasive does not mean exotic. So I just so we can get this clear, because this is something that bothers me. So you might have heard the term native plant and exotic plant. Native plant, and we can talk more about what that means later, but native plant means different things depending on which country you're talking about. But native plant generally means the plant that occurs naturally. Oh my gosh, I'm not typed today. Occurs naturally, but like for a long time, like for a very, very long time, over time, right? Like another thing you can call native plant is like indigenous. I don't think I'm spelling that right. Indigenous plant, native plant, indigenous plant. Exotic plant means a plant that originated from somewhere else, right? But then you have other terms: invasive plant and sorry, but this bothers me, invasive plant. Invasive plant is a plant that's aggressive and overtakes resources. A non-invasive plant is just plant that grows well in a community of plants, right? A lot of times people will say exotic and invasive and use those interchangeably. That drives me nuts because they're not the same. Native plant and exotic plant refer to where the plant occurred over time. Invasive plant versus non-invasive plant refer to how aggressive the plant is. So my point is, you can have native plants that are also invasive. You can have exotic plants that are also invasive. You can have native plants that are non-invasive, and you can have exotic plants that are non-invasive. So as a plant ecologist, this drives me nuts. I don't like to use these interchangeably because they mean different things. But anyways, both Azolla and Salvinia are problematic invasive species. Invasive species, meaning they are meaning they are aggressive and overtake resources in both their native and exotic ranges, both places. What do I mean by problematic? Here is a lake. Here is a lake that is covered with probably Salvinia. It's far away, but this is bad for the lake. This is bad. It's growing so aggressively that it is making It's blocking sunlight from entering the water. It's preventing gas exchange in the water. It's just not good for the lake. It's not good for any of the organisms growing in that lake. And some places you actually see signs saying that it's illegal. Look at this. Illegal to possess or transport these plants, right? You got to look at your boat before moving your boat to another place because it's such a problem, and it's bad. It's bad for the ecosystem. Sometimes people go to great lengths to remove it. Right here on the left, you see somebody with a boat, and they're dragging this floating boom behind it and kind of roping in the floating plants to then remove them. In other situations, people have tried to use this insect called a weevil to get rid of these plants, because the weevils will lay eggs on the plants and then the plants will be killed by the weevils. This is something that's called biological control. I don't know if you remember from the documentary. In that documentary, there was a biological control of using sheep to eat the knapweed, right? In that example, that probably works well because the sheep are not gonna like overtake, like probably somebody's there controlling the sheep, right? They're not just being let free into the world. In terms of the weevils, though, there can be problems with using insects in this way because they may attack other plants that are not problematic. Yeah, like goats to get rid of poison ivy. Actually, that's right, Rachel. And also, Rachel, goats are great to get rid of lots of things. Knotweed, Japanese knotweed, which is one of the more problematic invasive species throughout the Northeast. I think kudzu too, yeah. But goats eat anything, so goats are great. I wish we need more goats in the Northeast. Just like, let's put them along 95. Let's like have just goats going, like a goat lane. Let's have a goat lane. Goats can just eat along the sides of 95 and take care of all the invasive plants. Yeah, so unlike goats and sheep, which are normally being controlled by a human, there could be problems with using insects. Sometimes it works okay for a while, and then sometimes the insects decide they don't want to eat just this Salvinia, they want to eat something else that is not a problem, and then they could put that plant at risk. That happens. Okay, that's everything about fern. Now we've covered ferns. You know everything you need to know about ferns. You can look this over and the previous lecture over as you prepare for lab on Monday after the break, where you guys are going to cover ferns. Now we'll talk about the phenology assignment, which should be fun, should be a fun thing. So yay, phenology assignment. So this is an assignment that'll be, it says Thursday, the 8th, it should say the 10th, right? Because today's the 10th. Sorry, I forgot to change that. But starting today, Thursday the 10th, and due April 28th, the last day of class. The last day of class is when this is due. And this is all in the syllabus. You're going to do this phenology assignment. And the purpose of the phenology assignment is to get you outside. This is a beautiful time of year, usually, to get outside. And I know you guys are all stressed out and whatnot. And so I'm forcing you to leave your computers behind, leave your phones, like well, you should bring your phone because you're going to take pictures, but just get outside. Get outside. This last week, like I said, I was so stressed out, making myself sick. It's good to have time outdoors. And so I'm trying to make you do that. And also, this is a great way. Spring is so fun in this way because so many things change during spring. So this is a great way for you to learn about plants in our region as spring occurs, right? Longer day lengths, higher temperatures, rain instead of snow, right? Spring things. And just so you know, this assignment is going to be worth double the points of any of those regular homework assignments, right? So those regular homework assignments that are timed and stressful and not fun and are my attempt to get you ready to be doing very well on exams. This assignment is going to be worth 2 of those, right? So this is your opportunity to boost your grade if you're not doing so hot on those assignments. So when at the end of the semester, when you have 1 day left in the semester and you're like, I'm doing bad, how do I boost my grade? I'm telling you now, like, this is how. Do the phenology assignment. What is phenology first? First of all, what is phenology? So here's what trees look like right now. If you go outside, right? This is what they look like. Sticks. No leaves, no anything, right? Just boring sticks. However, pretty soon, this is what they're going to look like. And that's fun to watch. Believe me, it's super fun to watch. And it's something that, because we have very stressful lives that are full of our work and our family and everything else, a lot of times we overlook this transformation. You know, walking down, we're FaceTiming down the block, not even looking up, not looking around. And then next thing you know, you're FaceTiming down the block and behind you instead there's leaves, right? Well, now I'm encouraging you to look around, look up, because it's super fun to watch. But you don't just go from sticks to leaves, right? This takes time to occur because the plant has to grow the leaves. So this is your mission. This is your mission, is to document these changes. And there's a whole part of biology that is dedicated to this type of stuff, and that's called phenology. Phenology is the study of cyclic and seasonal natural phenomenon, especially in relation to climate and plant and animal life. And so this is your mission. This is what you're going to do. What are you going to do, right? What are you going to do? You're going to select 10 trees you encounter every day. And I want to really focus here on trees that you encounter every day, because believe it or not, you encounter probably hundreds of trees a day. You just don't maybe notice it. So select 10 trees that you encounter every single day. They could be around your neighborhood. They could be trees on campus. They could be near your work. They could be anywhere, near your kid's daycare. You know, involve your kid in this too. It's fun. Anywhere. Just make sure they're easy for you to get to, right? Don't take a weekend trip somewhere and pick 10 trees that are an hour away, right? No, pick trees that are close to your life. And just so you know, smaller trees are easier to observe more closely. You could also use woody shrubs. So anything right now that's like wooden sticks, right? It could be a tree or a shrub. So you're going to select your 10 trees. And on day 1 of this observation period, you're going to take photos, including a picture of you next to your tree, your trees. There's going to be 10 trees, a picture of you next to the tree, and then the tree's habit or form, and the tree's bark. Because since there's no leaves on the tree, the bark will help me identify what type of plant it is. That's on day 1. Over the next several weeks, you're going to revisit your 10 trees and take photos of any changes or any non-changes, right? If you go look at your trees 2 weeks from now and they look the same, you still need to document that. All right, so here's what I mean by what your photos should look like on day 1. So this is your tree. Look, this is your tree number 1. Oh, geez. Here's your tree number 1. Here's your tree number 2. And you're going to have a total of 10 trees, right? On day 1, you're going to take a picture of you next to your tree. It can be a selfie as well. If you don't have somebody with you, you can do a selfie. Just try to get, yeah, I'm sure you're all gorgeous And I'd love to see your pictures of your face, but just try to make sure you get some of the tree in the photo as well, right? Look, tree number 2, same thing. You next to the tree. You next to the tree. Your picture of you next to the tree, if it's not a selfie, will also show your tree's form. So if you have somebody else taking the photos of you on day 1, you can a lot of times capture the tree's form in the same photo of you next to the tree. But if you're doing a selfie, you may need to include 3 photos for the first day. 1 of you being a selfie next to your plant, 2, one that shows the form of the plant, and 3, one that shows the bark of your trees, right? So how many photos you need on day 1 will depend on if you're doing selfies or not. And you're going to revisit your trees at least once a week, Sometimes I get questions about, well, I'm going away on spring break, so how am I going to do this project? Okay, so today is Thursday. You could take photos today, or you could take photos tomorrow. And then if you go away for spring break, take photos when you get back. That's how that works. But at least once a week until April 14th, you're going to go visit your trees and take a photo and look at any changes. Look for any changes. It's okay if there's no changes, but you just need to document that. And what you're going to do is you'll have an Excel spreadsheet that lists your trees 1 through 10, the date of each photo, and the date of observed changes. So here, I'll show you what that could look like. Oh, before I show you what the Excel spreadsheet looked like, what are changes that you might see? You might see leaf or flower buds starting to emerge out of your branches. They might look something like this. It's okay if you don't know if it's a leaf or a flower bud. Like this, for example, is a flower bud. This, for example, is a flower bud. These are also flower buds, but leaves a lot of times will emerge from the same spot. So you can just, if you see something like this, you could just write as your observation, buds. Buds. You don't have to tell me if it's a leaf or a flower bud. You can just say buds. Sometimes it'll be more obvious, right? You'll see some beautiful flowers, and I hope some of the trees you select end up providing you this display, because it's very satisfying to be watching sticks for a few weeks and then all of a sudden your sticks are covered in flowers. It's pretty fun. Also fun is seeing new teeny tiny baby leaves emerging, which is another thing you'll probably see. Okay, and just I put the, okay, so yeah, the next slide has a lot of information. On Canvas, there's the full instructions are on Canvas, and there's an example of the Excel spreadsheet on that instructions. All right, so you're going to take photos of your trees until April 14th. That is the observation period from now until April 14th. You can do this as you go, or you can do this at the end, but basically you're going to have a 10-slide PowerPoint presentation where each slide has one of your trees, right? So your first slide would be all of your pictures for tree 1. The second slide would be all of your pictures for tree 2. And each slide should include at least 7 photos, 2 from day 1 or 3 from day 1 if you have to take a selfie, and then 5 from the subsequent visits. Even if no changes have occurred, you should still include a photo. This is very important. Back up your photos. You send them to yourself, make them in a Google Drive, because if you can't turn in photos that get lost. So you need to back up your photos as you go. Don't You can't write to me at the end of the semester and say your phone got stolen or your phone got broken and your photos are on your phone and you can't get to them. I'm telling you now, save them in a Google Drive folder, just in case, because there's nothing we can do if they're lost. So you're gonna take the photos until April 14th, and then 2 weeks later, you will submit to me this PowerPoint presentation. So you have 2 weeks to compile this PowerPoint presentation. Because the PowerPoint presentation, it will be a very large file, you have to save your PowerPoint presentation as a PDF for it to be able to be uploaded to Canvas. And so you will send me that PDF on the 28th. You can send it before the 28th. Remember, the observation period ends on the 14th. So you could send it anytime after the 14th. In addition to sending me those 10 slides, you'll also send me your Excel file. And your Excel file, see, example Excel file is on Canvas. I think it will be available, and I'll make sure that it's available when class is over. 10 rows, right? Trees 1 through 10, date of each photo, date of observed changes. You could see an example on Canvas. That's also due on the 28th. So on the 28th, you're turning into me one 10-slide PowerPoint presentation that's been saved as a PDF and one Excel file. And this is due by the 28th of April. No exceptions, no exceptions, no exceptions. You have 2 weeks after the observation period to submit. So no late assignments. Remember, this is worth double points. Okay. Might seem confusing. What I would say is pull up the instructions on Canvas when they're available, and I'll do that. Make sure they're available right after class is over. And if you have any questions, just send me an email or yeah. Right, so there's a question. The 2 photos from day 1 are of the whole tree and the bark, and then the 5 subsequent photos are just the tree. So for example, there's a couple ways you can do this, Michelle. So like, yes, on day 1, you have full tree with you next to it, or the full tree by itself, and then a selfie of you next to the tree and the bark. When you go on subsequent visits, the photos may look slightly different depending on what's going on. You might have a photo that's just like a close-up of one of the sticks. Maybe you showed up and one of the branches was starting to have buds on it. So like you just take a photo of the buds. You don't have to take a photo of the whole tree. If you took a photo of the whole tree, it might be hard to see the buds, right? So just take photos of anything that's changing. If nothing is changing, you can take a photo of the whole tree again. Or if there's like a really interesting display going on that's very observable from far away, like the entire tree is covered in flowers, fine. So your photo might look like the one on the bottom, or your photo might look like this, depending on, it's up to you. You can be creative. You can also include more than one photo each time you visit. So like, let's say you show up on one day and your plant has on it not only flowers, right, but also leaves too. Oh gosh, wow, 2 changes at once. You can take 2 photos that day and submit that. Just make sure you observe it steadily over the weeks. And you're going to include dates for all these things, right? So yeah, that'll help me pull it all apart, like to understand what's going on. Good question though, thank you. Any other questions about this? Like I said, I'll make sure that the instructions are on Canvas. Okay. Yep. Oh, it should say Canvas. Ooh. Not Blackboard, Canvas. Cool. So you guys are going to have a nice spring break. Enjoy it. Go outside. Have fun. when we get back, we'll start to talk about the seed plants. We're finally into the seed plants. There will be assignment now. What is the 15th? Hold on, let me just check. Oh, I think I changed this on Canvas. I think initially it was supposed to be due the 15th. I'm gonna make it be due after the break. So I'll double check the due date on. I think I'm having the assignment for which covers the ferns due the 21st, which is a Monday. So this is not right. Just to give you guys a little bit of a break. Is there any other questions? No? Okay, cool. Well, have a great break, you guys. And again, sorry for my camera, but I just think it would be better than having delays. But yeah, have a good break. If you have questions about any of this, send me an email. Yeah, see, Aya says, have a great spring break. You guys all have a great spring break. Professors, and we don't get a break. You guys get a break. I guess I get a break from teaching. Okay, fine, that's correct. But I'll be working all next week, unfortunately. You guys have fun though. You guys have a relaxing time. And yep. All right, I will see you after the break. Enjoy it. Bye, guys.